Hello, hi everyone. This time let's discuss about basic knowledge of toxicity and its terminology. So, when it comes to basic knowledge of toxicity, first of all, we should learn about what is toxicology. So, toxicology is nothing but it is a branch of science which deals with the study of poisonous effects of drugs, the study of toxic effects of drugs and other chemicals, and also with the detection, treatment, and prevention of toxic effect or of poisoning. This is the branch which is called as toxicology. Again, you see, toxicology is a branch of science which deals with the study of toxic effects of drugs and its chemicals, and also with the detection, treatment, and prevention of the poisoning. So in this, let's discuss about some terms and terminology. First one, you see adverse drug effects. So adverse drug effects. In the name itself, it is there adverse drug effects. When you take some drug, if any undesirable or unintended effect has been occurred in your body, so that is called as adverse drug effect. This is two types. It includes type A reactions and type B reactions. So type A reactions are also called as predictable reactions. It mainly occurs due to the pharmacological effect of the drug. And these are 80% of adverse drug reactions are type A reactions. Example, bleeding when you take warfarin drug, nausea when you take digoxin drug. And type B reactions, these type B reactions are totally unpredictable reactions. These are allergic reactions which occurs newly when your body responds to the drug. So these are type A and type B reactions. And next one, side effects. What are side effects? Side effects means it also comes under unwanted or unavoidable effects. When you take some therapeutic doses of drugs, unwanted and unavoidable effects occurs in your body. Such type of effects are called as side effects. Next, secondary effects. Secondary effects means also the same thing like side effects. These are indirect consequences or indirect effects when you take some drug. Next, toxic effects. Toxic in the sense poisonous effects. These mainly occurs due to low dose or prolonged use of the drugs. The toxicity tests are done for knowing the toxic effect of a drug. And what are those toxicity tests which are done? The first one, acute toxicity test is done for 14 days. Subacute toxicity test is done for 28 days. And subchronic toxicity test is done for 3 months. Chronic toxicity test is done for 6 months to 2 years. And special toxicity tests are done for carcinogenecity studies. It takes some longer period. Next, acute toxicity. What is acute toxicity? The toxicity which is produced by pharmaceutical drugs in one or more doses during a period which does not exceed 24 hours. So when you take a drug, within the 24 hours, if your body produces some toxicity because of the drug, such type of toxicity is called as acute toxicity. This test is done in each animal for one occasion only. For one time only, this drug is given to the animal and the responses are noted, and LD50 lethal dose 50 is determined. The symptoms of this test may include weight loss, muscle spasm, convulsions, lacrimation, diarrhea, and change in skin color. So what is the need for doing this test? The need for doing this test is to discover the unexpected effect of the drug. If there is any unexpected effect of the drug, that can be discovered by doing this test. And also by determining the LD50 of the drug, you can determine the lethal dose 50 of the drug. And then this information, the toxicity information helps in choosing the doses for clinical trials in humans. So this is why acute toxicity test is done. Next, subacute toxicity test. So subacute toxicity test involves it is an adverse effect which occurs due to repeated daily dosing of a chemical. If a patient is taking the same drug daily for over a period of several days or weeks, then for such type of drugs, subacute toxicity test is done. The testing period for such type of drugs is 2 to 4 weeks. and it mainly supports the initial clinical trials in humans. And the basic this evaluates the toxicity of a chemical. This subacute test evaluates the toxicity of a chemical, means how much toxic the chemical is can be known by performing this test. And also you can know the complete clinical chemistry of the drug by performing this subacute toxicity test. Next one, acute completed, subacute completed. The next test is chronic toxicity test. So it's chronic, in the name itself it is the chronic. Chronic means dangerous. So chronic toxicity test, it is an adverse effect which occurs after continuous administration of test sample for major part of the lifespan. For some diseases like HIV, cancer, like that, the drugs should be taken into the body throughout their lifespan. So that such type of drugs will provide chronic toxicity in the humans. For example, for rodents, at least 6 months duration is considered for testing the chronic toxicity. The endpoints are similar to the subchronic toxicity. The results and evaluation endpoints are similar with the subchronic toxicity. And this chronic toxicity is mainly done for longer duration of time. So that is why we have to select a large number of animals, as we have to do for longer duration of study, longer duration of time. So that is why we have to select the large number of animals for treatment. Then what are the objectives of this chronic toxicity study? The main objectives of this chronic toxicity study are to determine changes which occurs in the animal, and to determine the reproductive potential of the animal. Reproductive potential of the animals means whether they can produce their offsprings perfectly or not after using these drugs. And determining the damage of the fetus. When they are delivering a baby, if any damage occurs to the fetus because of these drugs, that also can be evaluated by performing this toxicity study. And also to determine the genetic impacts on the next generations. After taking these drugs, if there is any impact on the next offsprings or next generation of the animals, that can also be evaluated by performing this chronic toxicity study. Next thing, you see genotoxicity. Terminology, genotoxicity. What is genotoxicity? Genotoxicity is nothing but it is a destructive effect on a cell's genetic material which affects its integrity. In the cell's genetic material alone, it only affects its own integrity. Because of the genes will affect its own integrity. The human genes will affect its own human being. So that is what is genotoxicity. The toxicity because of the genes which are coming from your father generation. Some genes are there which are harmful to the offsprings. So such type of genes will come under genotoxicity. These are also comes under mutagens. Mutagens which causes mutation. Genotoxicity is also a mutation. Mutation means what? First of all, mutation is nothing but a sudden change in genetic field. Sudden change in genes is called as mutation. Here, a toxic change, the poisonous change is called as genotoxicity. The substance that has the property of genotoxicity is known as genotoxin. Genotoxic risk mainly includes this. Because of this genotoxicity in the offsprings, the next generations may get this cardiovascular diseases, cancer, multifactorial diseases like diabetes, and genetic diseases. So this is due to genotoxicity. Next one, you see carcinogenicity. What is carcinogenicity? It is the ability of a chemical to induce cancer tumors. The ability of a chemical to induce cancer tumors is called as carcinogenicity. The carcinogens may be of chemical substances, or it may be physical substances, or it may be biological agents also. Biological agents like virus, bacteria, fungi. Then when you consider the classification of carcinogens, there are mainly 3 categories of carcinogens. The first category of carcinogens mainly affects humans. These causes cancer in humans. And second category causes cancer in animals and also in the humans. And the third category, it does not cause cancer exactly, but it has a possibility of causing cancer. So that is third category. And the agents which are capable of causing damage to DNA, we have to know those agents. What are those agents? Mainly, electrophilic species can damage DNA, example, alkylating agents. And UV and ionizing radiations can damage DNA. Reactive oxygen species can damage DNA. Topoisomerase inhibitors can damage DNA. Nucleoside analogs can also damage DNA. And protein synthesis inhibitors can also damage this DNA. These are the agents which have capability to cause damage to the DNA. Next one, teratogenicity. So what is teratogenicity? Teratogenicity is nothing but it is a toxic thing which occurs and which causes the biological change, biological adverse effect on the reproductive system. So it causes some biological toxic effect on the reproductive system. So what is teratology? First of all, teratology is nothing but it is the science of studying, investigating the birth defects and their etiologies. If there is any defects in the birth of humans, the study of the birth defects is called as teratology. So the drugs, whatever the drugs, pharmaceutical drugs you take can affect the fetus during 3 stages. What are those stages? The first one is fertilization and implantation stage. During fertilization stage, means during after the conception of 17 days of a fetus, then unnoticed failure of pregnancy may occur. Abortion may occur because of these drugs in the first stage. And the second stage is organogenesis. When organs are developing for the fetus during 18 to 55 days of period, then also these drugs can damage those organs. And growth and development period means after 56 days, the organs are growing, the organs are developing. During that period also, when you take some type of drugs, it causes functional and developmental abnormalities of the organs. And what are the agents which can cause like that? Infectious agents includes viral infections, spirochetal infections like syphilis, and protozoal infections. These can cause this type of problems in fetus. And physical includes radiation. If any radiation is there, that radiation can cause this type of effects in fetus. Chemicals includes ACE inhibitors, like enalapril and captopril, these should not be taken. NSAIDs like diclofenac should not be taken when you are in pregnancy stage, and androgen hormones like estrogen should not be taken. So that is about teratogenicity. And the next one is mutagenicity. So what is mutagenicity? Mutagen, as I told you, mutagen induces the mutation. So mutagenicity is nothing but it is also a component of genotoxicity. It mainly changes the structure of DNA or structure of chromosome. And how it will changes is mainly by replacing the nitrogen in a base pair, or adding or deleting the base pair in a DNA molecule, or by changing the chromosomal number, or by mitotic recombinations, the changes in the genes may occur. So that is called as mutation or mutagenicity. So mutagen is a chemical which mainly induces this mutagenicity. Classes of mutations, there are mainly 2 classes of mutations. That is the first one, spontaneous class of mutation. Spontaneous class of mutation, what occurs is, it is caused during DNA replication. When DNA replication process is going, it may occur, or by incorporation of some incorrect nucleotide in the DNA change. By placing some incorrect nucleotide in the DNA chain also, it changes the DNA sequence, which comes under spontaneous class of mutation. Next, induced class of mutation. So induced class of mutation is nothing but the DNA is changed by environmental factors. Because of environmental factors, if the DNA structure is changed, that comes under induced mutation. Example, UV light can change the DNA structure. Next, types of mutations. There are mainly 3 types of mutations in that. The first one is chromosomal mutation. So chromosomal mutation means in the name itself it is there, changing the structure of the chromosome. It occurs in 4 stages. The first one is deletion. Deletion, due to breakage of the chromosome, some piece of the chromosome will be lost. That is called as deletion. Inversion. Inversion means sometimes the chromosomes breaks and again reattaches. That is called as inversion. Duplication. Duplication means nothing but it occurs when the same GNU sequence is repeated again, the duplication can occur. Next, translocation. The one part of the chromosome is transferred to another chromosome. These are not homologous chromosomes, but one part is transferred to another part. So that is called as translocation. Like this, by these four ways, the chromosomal mutation may occur. And also non-disjunction. Non-disjunction is nothing but the chromosomes cannot separate, they will not separate during meiosis process. So that is called as non-disjunction. And the second type of mutation is point mutation. So what is point mutation? A change in a single nucleotide. Single nucleotide sequence is changed during this type of mutation. Example, in sickle cell disease, it will happen, point mutation. And frame shift mutation. Frame shift mutation means what? The insertion or deleting of one or more nucleotides. The insertion or deletion, inserting or deleting the nucleotide sequence in nucleotides is called as frame shift. The shift, frame shift. So nothing but insertion, or you are inserting one nucleotide, the DNA sequence is inserting one nucleotide, or it is deleting one nucleotide. So that comes under frame shift mutation. So these are the three types of mutations: chromosomal mutation, point mutation, and frame shift mutation. So this is about basic terminology of toxicity.